It's published in the journal PLOS One, and it is on an axolotl. So usually, y'all, axolotls are described and studied because of their regenerative abilities, right? So you can remove limbs of an axolotl, they'll grow back. They even cannibalize one another. If you put them into a pool together, they will munch on each other, not for the purpose of killing, but since they regenerate parts of their bodies, it's like, hey, I need a snack. I'm going to snack on you out of their skin. Okay. That goo is a mucus membrane and it actually kills pathogens, right? So, and it has this small molecule in the goo called an AMP, an antimicrobial peptide. So, researchers in this paper were studying what exactly can these antimicrobial peptides do that are coming from the axolotl? What is it that they can target? The reason this is important is because antimicrobial peptides we don't actively use right now in killing bacteria or viruses or fungi or anything. And Melemony, right? We have an issue in this world of drug resistance and drug resistant pathogens like MRSA. And so this small molecule, the hope is that these small molecules can target other things that are toxic towards us. Does that make sense so far? Let me know. And yes, that's a huge thing, right? MRSA is a killer in hospitals. And yes, and it is terrifying as ferret mouth. And it worked against it. That's huge, number one. But Melemony, it also combated breast cancer cells. And Mel, where it gets even cooler, it wasn't because Melemony, right? You could just be like, well, Balint, did you just take these antimicrobial peptides and throw it on a bunch of cancer cells and they just happen to die? I mean, that's something, but it's not that impressive. So in this instance, when they did their cancer cell study, they didn't just take cancer cells and put them onto a dish and covered it with the antimicrobial peptides and see what happened. I mean, they did that too, but they knew Melemony was going to be a reviewer. And Melemony don't take no nonsense from anybody. All right. Melemony's just like, I don't do nonsense and shenanigansery here. I am a hardcore seasoned scientist, and I am not Rembrandt with a grenade. Or with science either. No, they do not have to kill the axolotl. No. And they're able to then synthesize the antimicrobial peptide as well. Yep, it is. So EOB Smikes, the the disease like virus, bacteria, fungus, it could be any of them, right, are evolving resistance against the antibiotics that we are making, and then have to make different ones, which then they evolve resistance to, and then vice versa. You just keep going. No, so the Red Queen, Mel, it's the idea, the hypothesis is based on Alice in Wonderland, where the Alice and the Red Queen are just running in place, and neither is catching up with the other. And it's because the argument is that both are evolving at the same rate. And so, because they're evolving at the same rate, neither catches up or takes out the other one. They just keep going. And they're just like essentially just for the antimicrobial peptides that they're identifying on these, again, they're getting the antimicrobial peptides through this gentle massage, as Smikesy said. Um, After they put it through this machine, they get the 22 potential candidates. And then Werewolf, they synthesize them. So they only needed to massage the axolotls at the start. Natasha, these are much harder to synthesize than other antibiotics that we utilize. They're just not as easy to make. And the reason is because of their chemical structure. So an antimicrobial peptide, right, has it's again a string of amino acids, but they have a positive charge to them, and that makes them water repellent. And so because they're water repellent and because they have a positive charge, they will bind to the negative charge of the outside of a bacterial cell wall, which has a slight negative charge to it. Because of this binding, they poke holes in the bacterium. and that causes cell death and leads to like irreparable cell damage and then the cell death. So that, as Smikes was saying, that there is this charge factor to it, and then making that charge is what causes this to happen. Difference between the male and female. So is there a sex difference? So yeah, it's not mentioned in the article. Yeah, so I didn't miss it. It's just not mentioned in the article. So werewolf, that would be an interesting question. It kind of makes sense that there's no difference because both sexes would need to be able to survive a potential infection. And so it would make sense that both would be able to then produce it. There might be at different levels, but there, and werewolf, you could also argue, well, there maybe would encounter different types of microbes depending on where they lay, possibly. So it could be really interesting to follow up with that. So they also looked at it against MRSA, like we talked about. MRSA is terrifying because there's so much drug resistance that it has, and they tested this small molecule on several different strains of MRSA, and what they found, so it was very, very effective against it. And the way they tested this was against the standard of care. So if you have MRSA, there is like this last ditch backup reserve antibiotic by the name of vancomycin. Vancomycin that they use. And this antimicrobial peptide worked as well, if not better. They suggested that it could be used as a combination therapy treatment as well for patients who need it. So it was a really good takeaway. They did suggest it would be really, really exciting to further adapt these peptides because in this study, they didn't change any of the peptides, right? They got the peptides out. They got the 22 of them. They synthesized them as they are. They tested them as they are. So if you can make them better, well, that would make a world of a difference, right? And that could very well happen to it. Yeah, so right now, Smikes, they synthesize not the whole mucus even, but just the peptides that they suspect have antimicrobial activity. Everything else, like that would lead to like the sticky goo substance, they don't need. They just are sequencing the peptides, which is also why it's cool that you don't need to massage the axolotls anymore. You don't have to put goo on your cells. You just put these small molecules in the syringe and it's good to go. I guess we do that. How does it target? Great question. So Smikes, with respect to the bacteria, so like MRSA, bacteria have a cell wall and it's slightly negatively charged. The antimicrobial peptide is slightly positively charged. So it binds, pokes a hole in it, and then that hole leads to cell damage, which leads to cell death. Only figure that we have in it. It selectively killed the breast cancer cells over the regular cells, which is, I think, quite remarkable. So instead of having anything else die, there was no other cell type that was destroyed. It just took out the particular cell type that was cancer cell and not the regular cell. That is not clear. That is not clear, Smikes. That is absolutely not clear yet if the axolotls are cancer resistant or not, because it's not clear if these small molecules act on their own cells, like the mechanism in the actual animal wasn't tested. They didn't test this on axolotl infections. They didn't test this on axolotl made tumor cell lines. None of it. And so that was something that needs to be tested downstream. It's to see, from that kind of perspective to it. Yeah, so that's what we don't know, nerd. Do we know if it's synthesized by the axolotl? It is synthesized by the axolotl gene tree. So they only found it. It was secreted from the skin in a mucus and not existing on the skin regularly. We know, Nerduino, how this mechanism works when it comes to killing bacteria. So when the bacteria are being taken out, it's because of a difference in charge, electric charge. But that isn't something with cancer cells, right? So the cancer cells and regular human cells, they don't have that like outside coat, right? They don't have that cell wall that would actually, or that barrier for like gram negative and positive that would be slightly negatively charged. Our cells don't have that. So then what's the mechanism by which these antimicrobial peptides are killing just the cancer cells? That's what we need to figure out. And Nerduino, what's really cool is in this study, they did not change around any of the signals. So they did not modify these peptides in any way. All they ended up doing was taking the peptides directly from the axolotl, synthesizing it as is, and then putting it on those cell types. And they found this activity. They did find that the MRSA killing or this resistant staph was as good as the last line of chemical defense that we have against it. So last line of defense that you would put on, for a person infected with staph, if you do that in a cell dish, the antimicrobial peptides work just as well. So they suggested maybe in order to be identified and like as a structure that needs to be, that the antimicrobial peptide can target it as being different, what stage of development of the tumor it needs to be, how far the progression is of it, how much you can dose. It's there's a lot of questions. There hasn't been anything tested in an animal. This has all been in a dish. But it's a really, really cool finding, especially coming from an axolotl, which I don't know. I was not. No, I don't think they were expecting an axolotl could have had this. Lunamon. Yeah, that's the big crux of this paper. They did measure gene expression in different cell lines after they gave the peptides on them. They even did different peptides, so they did peptide 1, 12, and 13 on those cells. Um, they did some genetics to compare what was going on when some peptides were put onto these cancer cells and found some really neat differences. Um, they were up and down regulated genes that were responding to the peptides, and that was helping with the fighting off of the cancer cell. Now, they didn't do anything after this, meaning that they didn't, get rid of certain genes or hyperactivate certain genes that they found from this candidate list. They just got a candidate list. These red bars here are what they consider as statistically significant. So above or below the red bars on either side here is what they consider important. They did not fulfill like the next step and actually test that, but that could be a future direction. That'll also probably actually take money. I mean, all this here, a mass spec and sequencing took money, but for them to continue on, they're going to need funding for it. And it's a really exciting bit of data, right? It's using natural products from an axolotl to treat disease, both in the bacterial front as well as on cancer in people. I think that's a really, really cool story. This is in addition to being already really cool because it's an axolotl.